So dear friends, in this video I will teach you about plant tissue culture, that is PTC, or in vitro plant cultivation. So in this video we will talk about definition, various requirements, steps in plant tissue cultures, how to measure growth in the petri dish or the suspension culture, what are the media requirements, what are the applications, and what is the growth curve. So developing of entire plants or organs or tissues or group of cell in vitro, that is in laboratory, using controlled aseptic conditions in a well defined media is called plant tissue culture. Let me repeat: cultivation of entire plant group of cell, that is tissue organ, using well defined media in a aseptic environment is called plant tissue culture. So what are the requirements of plant tissue culture? There is need of aseptic environment, that is environment fully free from microorganisms. Then we have to use some surface sterilizing agents to sterilize our explant, that is the starting material जिसके थ्रू हम शुरू करते हैं हमारा plant tissue culture. Then there will be sterilization room, the preparation room, aseptic cubicle, hot room, and general facilities like glasswares and other requirements will be there. So media preparation will be there, the introduction of the explant into media will be there, that is incubation, and type of tissue culture. Now there are two type of tissue culture: suspension culture, that is called liquid culture or fluid culture, or another one is static or callous or solid. So first we will see the definition of two terms, callus and explant. So for starting a tissue culture, we need the starting material from the existing plant. Suppose we want to do the tissue culture, suppose we want to raise an entire plant in the laboratory of neem or tulsi or atropa or belladonna or dhatura or something else, then we have to take the starting material from that particular plant only. The starting material is called explant, and this explant is a single cell which will give you entire organ, tissues, or entire plant. It is called explant. Then what is the surface sterilizing agent? There are few chemical agents which are used to sterilize the starting material. Directly after cutting the explant, we cannot use to start the tissue culture technique. We have to go for surface sterilization, that is we will just clean the surface using some bleaching agent or sodium or potassium hypochlorite solution or mercury chloride solution or hydrogen peroxide or some alcohol that are used in a fixed and a well defined manner to sterilize the starting material, that is explant. What do you mean by sterilization? To make something free from even a single living organism is called sterilization. Now second definition is callus. So what is the meaning of callus? After making explant from the existing plant, and we will keep this explant into some nutrient media, after few days there will be a specific type of growth. Your explant will be very bigger in size compared to initial explant, and this is called the callus. What is callus? The solid undifferentiated group of cell is called callus. There is no differentiation, all cells appear same in the callus, right? There is no differentiation like this is leaf or root or shoot or node. Callus is undifferentiated mass, cluster of the cells, that is called callus. Now what is the reason that each and every plant cell is able to make entire plant? So this is the beauty of the plant cell, that is even a single cell is able to reproduce into entire plant in aseptic condition with well defined media. So this property of a plant cell to become entire plant is called totipotency, and the cell is called totipotent cell, right? Now we will see two type of culture: static, that is solid or callus, and second is suspension or liquid culture. So static means there is no movement of the explant, it will be fixed in a petri dish in a solid agar media. On the other hand, in suspension culture, there will be continuous swirling or agitation using rotary shaker in an incubation manner. in the liquid media. So solid media is prepared into the petri dish for static media, and if we are going for suspension or liquid culture, then we have to use a special type of conical flask. They are called Erlenmeyer flask. In that flask, in liquid media, we keep 1 or 2 cells at either starting material, or we shift callus tissue into the suspension tissue, and this liquid media is continuously agitated in a rotary shaker which is having incubator features also. So in this technique, isolated piece of plant tissue is cultured on a nutrient medium. It is the culture of isolated cells or very small cell aggregates dispersed in glass liquid medium. The aggregates are obtained from the callus. The cell suspension is obtained by agitating pieces of callus in liquid medium on gyrating shaker. There is continuously round movement of the conical flask. There is a specific instrument called rotary shaker having some spring to fix the position of Erlenmeyer flask that continuously rotate because of the motor. And while rotating, there is no disturbances to the humidity, oxygen, temperature condition. It is maintained by the incubator. So it contains a uniform suspension of separate cells in a liquid medium. Callus liquid medium, agitated continuously, finally cells separated, and subculture of these cells are done. Cells are separated. After a periodical time, we will again remove the cell. We will shift to new Erlenmeyer flask, so that we will remove the accumulated waste product or the metabolite also, which we don't want. This can be achieved by rotary shaker attached in the incubator at a rate of 50 to 150 RPM, that is rounds per minute. Now there are 3 types of suspension culture: batch suspension culture, continuous culture. There are 2 types, open and close, and third is semi-continuous. So first is batch suspension culture, second is continuous. In that 2 types are there, open and close, and third one is called semi-continuous culture. So first we will talk about batch culture. These cultures are maintained continuously by propagating a small aliquot of inoculum in the moving liquid and transferring it to a fresh medium at regular intervals. You will remove a small liquid sample from the earlier batch and you will shift to a fresh medium, because periodically you have to supply fresh medium. Otherwise the used media or exhausted media will lead to degradation or complete spoil of the tissue culture technique. Generally, cell suspensions are grown in flasks of the capacity 100 to 250 ml, and they contain 25 to 27 ml of the culture medium. When the cell number in suspension culture is plotted against the time of incubation, a growth curve is obtained. So what is the growth curve? Growth curve is a typical type of graph which indicates the growth in the culture media, growth in the petri, or growth in the suspension culture with the time. So on the x-axis there is the time, on the y-axis growth pattern is there. So that is called growth curve. We will see later on in this video. A growth curve is obtained. Growth curve contains and based on the growth curve we can determine the measurement. So after the suspension culture is over, how do you realize, how do you measure the growth? Growth is happening or not? So what are the parameters to see the growth? You will compare the growth of the suspension culture or the cells compared to first day cell size. So in growth you will measure cell number, cell size, packed cell volume, weight and viability. So you will weigh the entire mass of the tissue. You will compare with the initial day, that is the first day. Viability, using some diet technique, you will analyze the nucleus or the organelles in the plant cells, whether they are viable, they are living cell, they are dead cell or living cells. Right now in packed cell volume, we will take the aliquot, newly formed cell, we will rotate in the centrifuge, then we will measure the packed cell volume. So these are the ways to measure the growth, whether growth is there or not. So there are few parameters. When the cell increases in the number, the liquid in the Erlenmeyer flask become turbid. So turbidity indicates cell numbers are increasing. As a result of more and more cell number, there will be more and more turbidity, and this turbidity can be measured using colorimeter. So more the turbidity, more is the newly formed cell. Now what is the viable cell taste? As I told you, to measure the viability of the cell, whether they are living or dead, there is some taste. So the staining methods such as fluorescein diacetate is used for accessing the cell viability. Dead cell appear as fluorescent red. So in this fluorescent diacetate staining technique, if cells are dead in microscope, they will appear red. Otherwise, they are living cells. So what are the applications of tissue culture? The first one is micropropagation. The asexual and rapid reproduction of various clones of a tree is called micropropagation, and this is the commercial. See, basically two types of applications are there for plant tissue culture. The first one is applications for commercial purposes and application for research. So the first and foremost, if we talk about commercial purposes, then there is micropropagation. Large production of same clone of same plant in minimum time period with less efforts is called micropropagation. That is the raising of similar clones in an aseptic environment using well defined media in an asexual manner is called micropropagation. Now, what are the research applications? There are many applications of plant tissue culture. Suppose you want to make one particular plant rich in some particular primary or secondary metabolite, then as per your desire, you can change the tissue culture media and you can design plants on your own as per your own criteria, right? So this is the main application. Then elimination of pathogen. You can produce some very strong virus or microbe resistant variety of plants by some genetic manipulation. This is elimination of pathogens, and you will produce hundreds of virus or disease free plants. Third is germplasm storage. That is, there are few endangered or extinct species. You can save their seeds or their germplasm. What is germplasm? The embryo or the seeds which can be stored for years in liquid nitrogen at a temperature of minus 190 or something around 190. This temperature is produced only in presence of liquid nitrogen, and this is the favorable temperature to store seeds or germplasm or embryo for a longer period of time. So then the production of haploids, different types of chromosomes in the plant, then artificial seeds, production of secondary metabolites. This is important from pharmacognosy or phytochemistry point of view. Suppose there is one plant, Atropa belladonna. So you get atropine from this plant. So you want excessive amount of atropine, then you may design plants according to your wish, or you can produce only that particular part of the plant which is rich in atropine. These are the applications of plant tissue culture. So important definition, just we have seen totipotency, callus. Now two more definitions we will see. Undifferentiated, that is, there is no differentiation in the organ or the tissue in the cell. That is callus. Already we discussed about it. What is meristematic tissue? The tissues which are rapidly dividing, rapidly growing, and generally very fresh, generally free from viruses, and generally present at the tip of the growing organ is called meristematic tissue. Already we have discussed micropropagation. And cleaned stock means the plants which are free from microorganisms that is prepared from the tissue culture technique only. So for orchids and nepenthes, these are the very used techniques to cultivate large quantity of orchid plant using plant tissue culture. So other application already we have seen. Then now we will see the definition of micropropagation once again. The asexual or vegetative propagation of whole plant using tissue culture technique is called micropropagation. So already we have seen, now we will see about continuous culture. We have seen earlier batch culture, that is at periodical time we will remove the aliquot, we will shift to new nutrient media. What is continuous culture? In this system, the liquid medium is continuously replaced by the fresh liquid medium to stabilize the physiological stage of growing cell. Cell proliferation takes place under constant condition. As a result, the active growth phase of the cell declines the depletion of the nutrient. The cell passing through outflowing medium are separated mechanically. The cells are always in the exponential growth phase. Cells will never be in a mode where their growth is limited, because continuously you are adding new fresh media. Now we will talk about culture medium. What is the media? Media is the plural, medium is the singular. So the culture medium is the medium which provide the growth material in liquid or solid form in terms of macronutrients, micronutrients, plant growth hormones or plant growth regulators or sugar or vitamins. So this composition is called culture medium. There are two ways. Either you can purchase ready-made tissue culture medium from the market, like for example Murashi's and Isku's medium, or you can customize based on experience or based on empirical way. You can design your own culture media in the laboratory, and there are only other available in the market. So a tissue culture medium basically contains these many minimum requirements. Solidifying agent, that is gelling agent, that is agar, to make the tissues, to make solidifying media. Then there is liquid for as a vehicle. Then there are macromolecules, there are micronutrients, there are vitamins, there are sugar and plant growth regulators. These are minimum requirements. There are some other customized requirements based on your research. For example, there is antibiotic also in some type of culture media. So we will see the macro element, that is nitrogen, NPK, nitrogen, potassium, calcium, magnesium and phosphorus and sulfur. They are the macronutrients required for tissue culture. Why macro? Because their concentration is more than 0.5 millimole per liter. If concentration in liquid media is less than 0.5 millimole, it will be called micronutrient, that is chlorine, iron, manganese, cobalt, copper, zinc and molybdenum. If we talk about their uses, then nitrogen is the source of proteins, nucleic acids, so it is requirement. Potassium regulate the osmotic potential. Calcium required for cell wall synthesis, because you are growing an entire plant which is available in the field in the laboratory, so you have to simulate all the conditions which is available in the actual outside our laboratory. For example, soil, water, moisture, then humidity, then dark and light cycle 12 hours each. That is to replicate the photosynthesis. That is the requirement. So if I talk about sulfur, it is component of some amino acids, phosphorus component of nucleic acid, enzyme cofactor, and lot many micronutrients are used as enzyme cofactor, right? Now, how to prepare a tissue culture media? It is the procedure for 1 liter. For example, add about 800 ml of distilled water to 2 liter flask. Then you have to add other contents. You have to check pH. It should be generally between 5.7. Add more distilled water to make the volume. You have to add agar and other media to make the tissue culture composition media. Then organic supplement, nitrogen in the form of amino acids like glutamine, then we have asparagine and nucleotide, then organic acids which are required for tricarboxylic acid. The acids required for citric acid cycle, we have to give like citrate, malate, fumarate and succinic acid. Then complex substances having more than 2, 3, 4 components in the mixture. Yeast extract, malt extract, coconut milk, protein, hydrolysate. So these are not required for all type of tissue culture. Depending on your need, depending on your output, depending on the plant you are using, and depending on the secondary metabolite or the disease you want to remove, you have to go for this type of permutation and combination. Activated charcoal is used where phenol-like compounds are a problem. Absorbing toxic pigments can be done using activated charcoal. We will talk about carbon source and vitamins. So source of glucose, sometimes fructose in the concentration of 2 to 5%. Most media contain myo-inositol, which improves cell growth. Then an absolute requirement for vitamin B1 is there. Thiamine can be used. Growth is also improved by addition of nicotinic acid and vitamin B6, that is pyridoxine. So some media contain pantothenic acid and benzoic acid. So already we have seen the applications of these macronutrients. Now we will talk about plant growth regulators. Basically, we know about auxins, cytokinins and gibberellins and abscisic acid. So auxins are basically used when we want the rooting, when we want to produce roots and don't want shoot, then there is the need of auxins. If we want cell division and shoot induction, then there is need of cytokinins. So zeatin is the naturally available cytokinins. Likewise, keratin is there, and gibberellins are basically for elongation of the internodes. Node means the area on the stem from which there is shooting out of branches or leaves. What is internode? The place, the distance between two nodes is called internode. Likewise, for induction of embryogenesis, there is need of abscisic acid. So, a very simple logic is there. If auxins and cytokinins are equal in concentration, you will get callus development, that is undifferentiated mass of tissue. You can't identify, it will become shoot, it will become root. On the other hand, if there is more concentration of auxin, less of cytokinin, you will get root development, and vice versa. That is, if auxin is less, cytokinin is more, you will get shoot development. This is simple logic. Now, plant regeneration categories. Enhanced release of auxiliary bud proliferation. There are two terms: organogenesis, that is the formation of individual organs, shoot, root, flower, leaf, either directly on the explants where a preformed meristem is lacking, or de novo origin from callus. Now, somatic embryogenesis is the formation of bipolar structure containing both shoot and root meristem, either directly from the explants or de novo origin from callus. Now, protoplast fusion is a technique where you fuse two cells by removing, by eliminating, by rupturing their cell wall using some enzymatic or electrical or mechanical ways. By this, you can have the best properties of two different type of cells. They may be of the same plant, different plant, same family, different family. It can be as distant as possible. You can take cell 1 of A plant, you can fuse with cell 1 of B plant to get the beauty of both the cells, both the plant in single cell. This is called protoplast fusion. Now, this is the most important and the last phase we will discuss. This is the growth curve. On x-axis you are saying the time, on left you are saying the growth pattern. So there are 5 phases: lag phase, exponential phase, linear phase, deceleration phase, and stationary phase. So lag phase is the initial starting phase in which the cell or the explant take their own time to acclimatize, to adjust, to set in the new environment, in the media. That is, cells prepare to divide. That is exponential. The rate of cell division is highest. Very speedily new cell divide, again divide, redivide, and there is huge growth of the good number of cells are there in the Erlenmeyer flask or in the solid, that is in the callus. Third is linear phase. Cell division slows, but the rate of cell expansion increases. Then there is fourth phase, deceleration phase. Rate of cell division and elongation decreases. There is no more formation of new cells, decreases and also decreases. And the fifth is stationary phase. Number and size of cells remain constant. No more growth is there. It means either you have to change the culture media, you have to add fresh culture media, you have to remove the fully mature cell from the earlier batch and you have to shift to new media, or you remove those fully mature cell and add new media for remaining cell. So in this way, we have seen the detailed summary of the plant tissue culture. Thank you very much.